So, good afternoon, everyone. Pleasure to be here. And hopefully I'll teach you a little bit about cytogenetics, how it pertains, at least in the clinical setting. And I kind of added these slides in the last couple of talks that I've given, and one in particular was a keynote talk at the Canadian Chromosome Conference, where you would expect people to get this answer correct. And about, I think, 25% of the audience, it was about 160 people, got this correct. So I'm going to pose this to you. How long have we been looking at chromosomes? And I'll make it a little bit easier. Let's break it up into A, B, and C. So how many of you think A? I preface, okay, B, C. We're kind of equally split between this crowd. Surprisingly, we've been looking at chromosomes for about 140 years. That's pretty remarkable, and that actually comes from Strasburger in 1875, looking at this in plants, and then a little bit later by Fleming, looking at it in animals. And so it's pretty puzzling, if we've been looking at chromosomes for over 140 years, why it took until 1956 for us to get the human chromosome number correct. I mean, prior to that, we believed that humans had 48 chromosomes. Anyone have any ideas? Well, I mean, why would it take us so long, to kind of get that right? The answer is that prior to 1956, it was technically a big challenge to study chromosomes. Very, very difficult. It was hard to get dividing cells, and when you did, the chromosomes were most often just kind of sprawled all over each other in clumps. They weren't spread out. And so it was really the advances that came in the 50s, 60s, and 70s that helped us kind of get to figure out the correct chromosome number. And once we could study human chromosomes, we started seeing the associations with certain clinical entities like Down syndrome, etc. But these are the primary things that helped us. The mitogens, basically pushing the cell into active cell division. Once we get it in active cell division, to arrest it at the point where the chromosomes are visible in metaphase. Hypertonic solution to spread them out. That's getting a bad echo. Hypertonic solution to spread them out. And then the ability to differentiate each individual chromosome so that we could figure out what is going on. So I stole this from my old mentor, Kurt Hirschhorn, who is down at Mount Sinai. And Kurt was, again, one of the huge people involved in early clinical cytogenetics. And this is what a metaphase would have looked like in the 60s. Again, this would have been using some of the advances of spreading out using hypertonic solution, but you can clearly see that to be able to distinguish that chromosome from that is very difficult. And it was really the differential staining technologies that I'm showing you over here that helped us give kind of individual signatures to each of the chromosomes. So this is cytogenetics in the 1950s and 60s. Any of you looking at old textbooks would have seen they used to group them according to size and the position of the centromere. That's group A, that's group B, there's C, and D, etc. And they often used to refer to group E chromosomes as, like Down syndrome, the additional ones. And so that's kind of what it looked like then, and this is what it looks like now. And this is with the advent of G-banded staining, which is the primary the predominant way that we look at chromosomes and stain them. And you can see unique banding patterns. You may think of them as barcodes, but each chromosome has a specific banding pattern. And once you look at them long enough, they kind of be like your kids. You can point them out anytime. They can be in any shape, any metaphase. You know what's there. And hopefully you get to know what's missing and what's extra, which we know is associated with pathology in many cases. And so, taking you through the journey rapidly, as I've gone from 1875, we get into the late 1970s and early 1980s, where you had a marriage, really, of molecular techniques and cytogenetic techniques in the form of molecular cytogenetics. And it's really the application of these molecular biology cytogenetic preparations that took us to the next step in clinical testing. And so molecular cytogenetic techniques have provided a way for us to detect complicated, cryptic, and submicroscopic rearrangement that remain undetected or undecipherable. So what you'll see is molecular cytogenetics gives us a way to figure out things that you cannot see, but it also helps you to figure out things that you can see but just don't know what it means. The hallmark of molecular cytogenetics was actually FISH, fluorescence in situ hybridization, where you take a DNA segment of interest, you put a little fluorescent probe onto it, and then you're going to basically hybridize it to chromosomes of interest. And just taking you through what those steps would be, here you see in the cartoon the DNA, which would be really the DNA that sits in the form of a chromosome sitting on a slide. And in all types of molecular biology, you have to be able to have access to the DNA, so you're going to generate single-stranded DNA by denaturing it, typically by heat and a combination of chemicals. You're going to add your DNA probe, which is a small fragment. It has a little fluorescence signal on it, and that probe, in most cases, started off as a double-stranded probe, and you're also going to denature it, make it a single-stranded. It's going to then find its complementary sequences and then hybridize or stick to it. And then things are going to cool down. You're going to look down the microscope and just kind of look for your green dots, your red dots, or whatever. But you know that that green dot or red dot is associated with your specific probe that you have chosen. So these are some of the types of probes that we use in clinical testing. They're the beta satellites that you see over there. There are the alpha satellites, which are synonymous with the centromere probes. We often use these for counting the number of chromosomes. They're nice, big, bright probes, and they're very preferentially utilized. These are the classical satellites that you see. And the ends of the chromosomes, the telomeric sequences, they're not truly telomeric sequences. They're, in fact, subtelomeric sequences because the telomeres of all our chromosomes have the same repeating sequence. So if you want to be able to identify a specific chromosome, you have to go to a region that is unique for that specific chromosome, and that's going to be just underneath those repeated sequences. You'll often see them referred to as telomeric probes or subtelomeric probes. They're the same thing. Then obviously we want to be able to pick up specific and unique regions in the genome, and we can paint a whole chromosome arm, and we can paint the whole chromosome. So FISH becomes really nice and pretty, as you'll see some nice slides. This is an example of utilizing FISH in cancer. I'm going to show you only a couple of examples. Most of today is going to focus on just constitutional genetics. But this is an example of using a specific probe for the c-MYC region, which is on the long arm of chromosome 4 at position 2,4. And there you see the FISH probe for c-MYC lighting up in its endogenous natural place. But what you recognize on that chromosome is a huge amplification. c-MYC is an oncogene, and we know you can get significant amplifications, in this case, in AML. This is just a nice picture to illustrate whole chromosome paints. And in this case, it's been very informative to help us delineate a three-way translocation. You can actually appreciate just by virtue of the colors. You don't even need to be a cytogeneticist. You just need to know that, chromosome 1 is in green, chromosome 4 is in orange, and chromosome 7 is in red. And there you can clearly see that chromosome 1 and chromosome 4, there's chromosome 7, and you can see how chromosome 4 over there is sitting on top of 1. Chromosome 7 has been translocated to chromosome 4, and chromosome 1 to 7. From a clinical point of view, we know that FISH on interphase nuclei are particularly useful in certain clinical situations. We can look at interphase nuclei that are derived from chorionic villi or amniocytes, so in a prenatal setting, we can look at these types of nuclei. In the past, people have looked at, in a pre-implantation genetic diagnosis setting, an IVF setting, we've looked at interphase nuclei, particularly in the blastomeres, and in the absence of available metaphases in various cancers. So in certain cancers, it's hard to get dividing cells. You don't get a lot of cells, but you will always get, in many cases, the actual nucleus that it hasn't divided, and you go straight and look at those. I'm going to come back to this a little bit later, showing you now it's not really standard of care to use FISH for PGD, and that's for very specific reasons. What's the big difference between metaphase FISH and interphase FISH? Well, here's the metaphase, and let's show you chromosome 18 hybridizing. That's the X chromosome in blue, chromosome 13 in red, chromosome 21 in green, the Y chromosome in gold. So you could recognize that it's a male either by the fish or by virtue of the fact that you can actually see the X chromosome by its band, and there you can see the Y chromosome by its banding. What does that look like in an interphase nucleus? Well, there's no difference in the DNA and the actual sequence. The only difference is how you visualize that. And in an interphase nucleus, it's just going to be like that. So you don't see distinct chromosomes. You're going to see the actual fish probes. And then again, very easy. You just need to know which fish probe matches with which color, and you can easily see that this is a male. Whereas in the previous one, you could do it by banding. This, you really require fish to help you do that. This is an actual a case from a prenatal diagnosis. You're looking at an interphase amniocyte. So these are 4 specific amniocytes. The probes that we're using in this case are the X chromosome. So immediately we know this is a female. Looking at chromosome 21, you see two copies of this. And we know that this is disomy 21, meaning 2 copies of chromosome 21. When we do that, we don't know anything else. This child could have cystic fibrosis. That doesn't tell us anything other than there are apparently 2 copies of chromosome 21. And the one on the right, there you see the X chromosome, a single copy of the Y, but unfortunately 3 copies of chromosome 21. The X and the Y tell us it's male, and the 3 signals in each nucleus tells us that there are 3 copies of chromosome 21. That's a prenatal diagnosis of a male with Down syndrome. So very easy and very quick for us to do that. How good is FISH in a prenatal setting? And there was a really good study coming from the LabCorp group, actually as part of a consortium in 2001. And what you can see looking at the sensitivity, outstanding positive, negative predictive values, very, very, very high sensitivities and accuracy using FISH in a prenatal setting. The study looked at, their study particularly looked at over 5,000 pregnancies, and when they calculated their statistics, they incorporated over 30,000 additional pregnancies. So really a good indication of its clear utility in a prenatal setting. So what are the common indications for prenatal interphase FISH? Advanced maternal age. We know age is highly correlated with aneuploidy, and as you get increase in the maternal, so you get an increased risk for aneuploidy, so that's a very common indication. You can get an abnormal maternal serum screen indicating an increased risk for trisomy 18 or 21, or an abnormal maternal serial AFP, which indicates an increased risk for neural tube defects, which in some cases is also associated with a chromosome abnormality. And then, obviously, if there are prenatal ultrasound abnormalities, that's another indication of a potential chromosome abnormality. What are the benefits of prenatal interphase FISH? Well, we know that the trisomies 13, 18, 21, and monosomy X are the most common aneuploidies related to maternal age and or fetal abnormality. Routine chromosome testing takes about 7 to 10 days. The reason is we have to culture those cells. We have to get them growing. We have to get enough of them. Then we have to stop them in metaphase, band them, etc. And that takes about 7 to 10 days. Interphase FISH gives us, therefore, a rapid way to screen for the common aneuploidies in uncultured amniotic fluid cells or CVS cells in about 1 to 2 days. Practically, we can even do this in about 4 or 5 hours. Technically, we can do it in 4 or 5 hours. Practically, by the time the obstetrician does the procedure, to the time it gets to the lab, to the time, it's at 5 o'clock in the evening, it's generally going to take, in most cases, a turnaround of about 24 hours, and in some cases, 2 days. And if it's on a Friday, then it's going to be on the Monday. What this does allow, what it does give us is the opportunity to reduce emotional burden on the patient, and very often on the physician who has to deal with these tough situations with their patients once they have an increased risk for chromosome abnormalities following an abnormal screening result. It provides an opportunity to reduce anxiety through earlier decision making. What are the limitations? Well, as I said, it's very quick. But it has reduced sensitivity compared to conventional cytogenetic analysis. So cases with cytogenetic abnormalities, other than the most frequent abnormalities, will not be identified by this technique. You're looking for very specific things. The fish can be normal. You could still have an unbalanced translocation involving other chromosomes. You could even have an abnormality of chromosome 21 in a region not covered by your fish probe. So it's, the most common things are the things that you're going to target using FISH, but it does not give you a clean bill of health. It's very specific. Showing you what this looks like in an IVF setting, and this is actually looking at an embryo, a blastomere. And here we're looking at chromosomes 13, 16, 18, 21, and 22. There you see 2 copies of chromosome 18, 2 copies of chromosome 13, 2 copies of 16, 2 of 21, but unfortunately 3 copies of chromosome 22. Trisomy 22, we know if you put that embryo back, it is going to miscarry. It is lethal. Trisomy 22 is the second most common chromosome abnormality that we see in miscarriage specimens. So, as I alluded to right in the beginning, this type of technology in an IVF setting for pre-implantation genetic diagnosis has now been substituted for the newer technologies, and I'm going to show you a little bit later. And you can ask why. I've just showed you how good the prenatal FISH is. Outstanding positive and negative predictive values, outstanding sensitivities and accuracies. So why is it? And the answer is by virtue of the fact that you're looking at one cell. You take one cell, if there's a little bit of dust or debris, it can affect the way those probes are hybridizing. You have to make an answer, give an answer based on one cell. And there's a lot of subjectivity associated with that, as well as the technical issues that can affect your ability to make that interpretation. And so it has been, it was good for some patients, but overall, in prospective studies that looked at the benefits associated with IVF PGD FISH, they have not been recognized. And I can, thankfully say that the newer technologies are giving us that, and I'll show you that a little bit later. So FISH is no longer the standard of care for PGD. Moving into a pediatric setting, the microdeletion syndromes, and this is where FISH had one of its greatest utilities early on. Reminding you, and I believe you are going to have more extensive lectures on microdeletion syndromes. So the microdeletion syndromes are deletions of a megabase or so of DNA that are most often too small to be seen under the microscope. So most of the time, if we look at the chromosomes, they look normal, but there is something smaller deleted that we cannot appreciate just by visual inspection. These microdeletion syndromes produce well-defined contiguous gene syndromes, which are characterized by multiple apparently unrelated clinical features. They were originally defined by high-resolution banding, and now most are diagnosed using molecular cytogenetic techniques. Here are some of the common cast of characters that we see. DeGeorge syndrome that comes with the about 75% of cases having cardiac defects or very specific cardiac defects, about a quarter of them having schizophrenia presenting later on in life. And you can go through the list, and as I said, I think you're going to have more extensive lectures on these subjects. But in a clinical laboratory setting, we rely heavily on our clinicians to tell us what FISH test to do. So when the clinician sees a patient like this with the accompanying cognitive profile that really is suggestive of Prader-Willi syndrome, they would then draw blood, send it to the lab, and we would run the Prader-Willi test. And in this case, you see there's a deletion. Over there, the probe looks for its matching sequence. It's not there. It can't hybridize to anything. Therefore, you do not see a signal. And so you see a clear absence of the Prader-Willi FISH probe confirming the diagnosis. In this case, again, together with the whole cognitive and developmental profile of this patient, the clinician may be thinking of something like Williams syndrome. They would order the Williams probe, FISH, and there we can clearly confirm that diagnosis. But what happens is you get these types of patients that have this whole gestalt of a chromosome abnormality. They have intellectual disability. They are dysmorphic. They have that whole facial features that suggest a chromosome abnormality, but they don't look like a Prader-Willi, or they don't look like any of those microdeletion syndromes that I showed you on that long list, which is always expanding. And so the question is, what FISH test do you do here? You can try one and then it's negative and try another, but ultimately it becomes, as I refer to it, as a fishing expedition, excuse the pun, and it becomes expensive, and in most times you're still left with no diagnosis. Bear that in mind because I'm setting up the scenarios for the need for new technologies. This is a second scenario. This is a patient with multiple congenital abnormalities. We look at chromosome 5. clearly there is additional material. You can see that's the normal, that's additional. We look at that, there's not enough banding information for us to understand where that comes from. And so again, how can we figure this out? We can do multiple paints, and here we see the answer. It is chromosome 5, and there's that abnormal one, clearly with the additional piece deriving from chromosome 11. But the question you should ask me is, how did I know to use an 11 paint? And the answer is we didn't. We started with 1, it was -2, -3, and ultimately it's going to light up, but it can be expensive, long, etc., etc. Another similar situation is that of a marker chromosome. A marker chromosome is really a chromosomal fragment that does not have enough information for us to define where it comes from, and we just refer to it as a marker chromosome. So how do we identify it? Again, we can go through similar options, sequential trial and error with all these paints and probes, and then ultimately we'll get the right answer. So I spoke about the need for new technologies. It came from all these situations where you have that child that has normal chromosomes, but clinically has a gestalt of a chromosome abnormality. Where you get these small pieces, you don't know where they come from, where you want to look for a whole bunch of things, either that you can see, but don't know where it comes from, or that you can't see and figure something else is going on. And so, as I said, there has to be an easier way, and there's multiple different things that came in between. I'm going to take you very quickly through the journey from straightforward fish all the way to our microarrays. But this is kind of some of the evolution that you saw using reverse fish, multicolor fish. That's like a PCR. Brynn's is like a PCR directly on the slide. Fiber fish, very good for ordering specific probes. You stretch out DNA into long fibers. And then chromosomal CGH, which then sparked the whole microarray era. So I'm going to just concentrate on 2 of these now, multicolor fish and then DNA microarrays. Multicolor fish, an outstanding tool for primarily in cancer. Why? Because in cancer, we very often see complex rearrangements. Whole fragments rearranged, and if a dark band goes somewhere else and it takes the place of another dark band, how can you see it? How can you know where it's gone? Or if it's very similar material translocating somewhere else, it's very difficult to understand these complex rearrangements. And here you can see just by getting the chromosomes in different colors, it's a complicated methodology, but here you can clearly see that that part of chromosome 1 inserted in the top right comes from chromosome 14, and there you can see chromosome 2 translocated to chromosome 22, giving you an idea of some of the more complicated cancer cases. There you can appreciate these very complex, well, in cancer there's many more than 46 chromosomes often, and so it's quite a mess to kind of sort this out, and this is where the spectral karyotyping, which is what SKY stands for, it's the same type of concept of multicolor fish, has been very useful. So I'm going to leap very quickly to microarrays, and think of microarrays as being a multiplex fish test. So instead of just doing one, we're doing a couple, and ultimately we're doing millions of probes all at the same time. And so we really jumped from, having something great to something being absolutely incredible. So in this illustration, by fish, we would take a DNA segment of interest, as shown by that kind of green bar, and then we would hybridize it back, and we'd look for the signal. And now what we do is we take these representative regions of the genome, and we spot them down onto a microarray. I'm going to show you how it's done in a while, but what is the main benefit of doing fish, of doing a microarray? Remember, fish gives us things that we can't see, and so will microarrays. So it comes down to banding resolution, which I'll delineate in a couple of seconds. But the resolution of cytogenetics is round about 5 to 10 megabases. And that's really about one band or so. Once you get below that, it becomes very difficult to see gains and losses. And microarray is going to really increase that in the order of 1 to 5,000. I mean, significant improvements in the resolution. So what are the limits of chromosome testing? It gives us a visual examination of the whole genome. We have the entire genome in front of us, but it gives us outstanding coverage, but it does not give us the best resolution. What do we expect when we do cytogenetic G-banding, karyotype? We should see obvious gains and losses and rearrangements, but what we can appreciate is that the smaller the region of gain or loss, the harder it is to detect. And at absolute best, imbalances in the realm of 3 to 5 megabases, and that's really almost theoretical, may be detected. In reality, you're going to see changes of about 5 to 10 megabases and often a little bit greater. And absolute best is dependent on banding resolution, and that in fact can be influenced by whether you're looking at a miscarriage specimen, a cancer specimen, a blood specimen, an amnio specimen, etc. So a normal karyotype does not rule out, obviously, things that you're not looking for. As I said, you have normal karyotype, you can still have Huntington's disease, cystic fibrosis. So single genes are another category that you would have to do in terms of a separate analysis. It is not a mutation assay, but a coarse whole genome analysis. A normal karyotype does not rule out small and subtle gains or losses and rearrangements. Why use microarrays? Well, its clinical utility in constitutional and cancer cytogenetics has really been well documented in pediatrics, in prenatal and adult studies, as I'll show you a little bit later, as well as in IVF PGD setting and in a cancer setting. And it's been a phenomenal tool in cancer research as a mechanism to identify candidate genes that cause and initiate the progression of certain tumors. When you use certain microarrays, your choice is determined whether you want to do clinical testing or whether you want to do research. And when you're considering those two things, you should be thinking about probe spacing and resolution, as well as genotype information, and I'll elaborate on those things as we go on. So let's tackle resolution and spacing. And in these illustrations, the purple bar is going to be your patient's DNA, and the green are going to be those probes that I showed you speckled down onto the slide on the microarray. And in these examples, you have 3 probes there, head to tail, 100 kilobases in size. There you have another 3 probes, 100 kilobases in size. And in this example, they're separated by about 1 megabase. In the normal situation, just like in fish, you're going to get hybridization of the patient DNA to the probes, as shown over here. You have a laser that's going to look for those probes, and in this case, the arrows are telling you that it's detected them, report back it's there, and give you the thumbs up. If you have a deletion, as shown on the left, those 3 probes will look for their matching sequence. They're not going to find it. The laser is going to report to you that there's a deletion. Now, if you have a deletion that is smaller than the size of your probe, so here you see a 30 kilobase deletion, but your probe is 100 kilobases. What do you think is going to happen there? Okay. That probe is still going to hybridize kind of to the left and right. Your laser is still going to recognize that that probe is there. And so you have to have the gain or loss to be far bigger than the size of your actual probes. So this was some of the issues that we dealt with in the original designs of the microarrays. And I'll show you how what people did was that they said, okay, let's take that 100 kilobase region and make much smaller probes. And this led to the development of what we call the oligonucleotide arrays. And oligonucleotides are typically about 25 to 50 base pairs in size, and you can space them, as far apart as you want. And so now you get significantly more coverage over that specific region. And so now, for our 30 kilobase deletion, you do have probes that should go over that region. And while those 2 big arrows are finding that, everything to the right, you should have the laser looking for those specific probes, will not find them, report back to you that they're not there, and you'll pick up that small deletion. And this is what I refer to when I'm talking about resolution. How small of a gain or a loss you can detect. Obviously, spacing, if you don't have probes in a specific region, no matter how good they are left and right of that, you're going to miss that. Okay, so where you put your probes, how big the individual probe is, and how far apart they are spaced from the next probe are very important in your ability to detect gains and losses. So the original microarrays were these bacterial artificial chromosome arrays that were used primarily to detect gains and losses of about 100 to 200 kilobases. This followed up, as I said, with the oligonucleotide arrays, showing that you could get much smaller gains and losses, and these can potentially detect gains and losses. I mean, actually, it's unlimited, from 100 to 100 kilobases and larger, but remember, each probe is about 25 to 50 base pairs in size, so it depends where you put them in the design of your array. In most clinical testing, you're going to be talking about 50 to 100 kilobases and larger, but potentially you can pick up things far smaller. Different designs targeted are going to be used to interrogate specific genomic regions of known clinical significance, like the microdeletions. They usually cover the telomeric and pericentromeric regions. And then the whole genome tiled arrays. These arrays are spread across the genome at various intervals. And as I'll show you, they may reveal gains and losses that are not clinically significant and may just be normal variation. And we'll speak about that. So this shows you one of the original CGH microarrays. You would have representative DNA sequences from each chromosome that were cloned into these backs. You would spot those down on a microarray, and then you would use a patient genomic DNA in one color and your control in another color. You would hybridize them, and the concept is if they had the same amount of DNA, you would see an equal mix of hybridization, which would be reflected in kind of this orangey color. If your patient had a deletion, you would have more of the control hybridizing, which would be seen as a greater of the red color, and if your patient had a gain, they'd have more of their DNA hybridizing, which would be seen as a greater green intense region. So that's why it was called comparative genomic hybridization, because you have your probes on the microarray, and then when you do the hybridization, you are hybridizing two different genomes, one being your patient, one being a control, and of different colors, and you're looking at the intensity. And you can see in this case, wherever there's greater green intensity, it's a gain of that specific probe because there's more of your patient hybridizing there. This is what one of the original CGH microarrays looked like. Normal for chromosome 21, and what you're looking at is the log2 ratio. Log2 of 0 indicates a copy number of 2. And there you can see the log2 ratio shifting up. And what they used to do in the original CGH arrays is they used to run this experiment twice. One where the patient was in green and the control was in red, and then one where the patient was in red and the control in green. And you'd expect to see when the patient was in one color a positive shift in the log2 ratio, or when the patient was in the other color, a negative shift. And when they saw the splaying, they knew they had the right answer, that it was an absolute gain or loss depending on which way the log2 ratio went. But have a look here at the number of probes in some of the original backs. You can count each dot. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12 over chromosome 21. Now, on the arrays that I use, or that we use at Columbia and most places, we probably have 100,000 probes over chromosome 21. And that's something like that. I mean, the order of magnitude of the number of probes that we utilize is just completely different. But this is what set the scene. So think of 5 basic diagnostic categories: normal, and this turned out to be a little bit more difficult than we thought it would be when we started using these new technologies. You obviously want to pick up Down syndrome. Then you want to go way beyond that, at that kind of crux of the resolution, to pick up your markers, your unbalanced rearrangements. You want to pick up all your microdeletions, your cryptic unbalanced translocations. You want to define new microdeletion syndromes and go all the way down to the gene level, and I'll show you some examples. And then the last category really brought a new level of diagnostics to cytogenetics, was integrating SNPs. Gives you the ability to pick up uniparental disomy, triploidy, consanguinity, mosaicism, MCCs, maternal cell contamination, and parent of origin. And what I call leveraging the genotype information from SNP microarrays for cytogenetic purposes. So I spoke about normal being a little bit tough, and in the beginning we started seeing that everyone had a gain or a loss. And so I know that what we know now is all of us have deletions and duplications in our genome, every one of us. And the majority of these are really what's part of our normal variation. The hard part is, when does a deletion or a duplication become pathogenic, causing disease? There is a general trend that the smaller the deletion or the duplication, the more likely it is to be benign, particularly when it doesn't cover genes that are significant. and the larger it is, the more likely it is to be pathogenic. But the gene content really has to be taken into consideration, because you can have a deletion of one gene that can give you, in the Duchenne's gene, and can give you Duchenne's muscular dystrophy. So these are what we refer to as copy number variations. There's different frequencies in different populations. How do we minimize detection of benign CNVs when we do clinical testing? Well, you can restrict the analysis to regions that are known to have clinical significance, like your microdeletion syndromes. Everywhere else, which we refer to as the backbone, you can choose certain size cutoffs. We started off using 200 kilobase for deletions and 500 kilobases for losses. And a lot of this has to do with the normal variation literature that came out, showed that all of the gains and losses that all of us have tend to be very small. They tend to be way under 100 kilobases, the majority of them being 1 to 20 or 30 kilobases in size. So as a general, we started off, but what you'll see is across the country and across the world, these backbone cutoffs differ considerably across all the clinical labs. And that is actually still an issue. Category 2, aneuploidy. So this is looking at a SNP array, and each dot is a probe, and how you can see significant. This was one of the first one we used that have 500,000 probes, and you don't have to be a cytogeneticist to see something's different there. That's elevated in the intensity, and that's chromosome 13. That's trisomy 13. Again, that's trisomy 21. Very easy diagnosis to make by an array, and we didn't think this would be an issue. But the partial aneuploidies is what we were looking for, and it's really been great for helping us to characterize genomic makeup of unbalanced regions, uncovering complex nature and possible mechanisms. And I'm going to take you through a little kind of diagnostic odyssey of how we used to do things and how things are done now. So here's your marker chromosome. Remember your unidentified flying little chromosomal fragment. We don't know where it comes from. And the outcome can be anywhere from benign to really severe. And so we want to get a handle on where does this little marker come from. The old way, we used to say that the majority of markers come from the acrocentric chromosomes, and the majority of those come from chromosome 15. So we would do a chromosome 15 fish. And there are the two normal 15s. There's the marker. It doesn't light up, so we know it's not from 15. Well, what about the other acrocentrics? How about 14 and 20, 13 and 21? They share homology. So we can see there's two 13s, two 21s. The marker doesn't light up. Okay, what about the next acrocentric pair, which is 14 and 22, that also share homology? And bingo, now we've done, I've lost count, how many probes? We've done a whole bunch of probes, and all we know is that it's either from 14 or 22. So let's do a paint for 14. We do a paint for 14. It's negative, and we pray that when we do the 22 paint, it should light up, because that's, and it does. Okay, so things are working well. We've just done a lot of fish probes, and all we know is that it's from 22. But how much of 22? Because as I said, depending on how much, can be benign or give us the cat-eye syndrome. So we can then run additional unique sequence probes. In this case, we're running probes that extend to the aryl sulfatase A gene and the DeGeorge region, which is just underneath the centromere, and there you see it lights up. So all we know is that it's at least to that specific probe. We do a microarray, and we obviate all those long fish things, and in one shot, we get everything. And just to orientate you here, this is the software output. It differs depending on what software you're looking at, but there's the log-to ratio, and there you see a shift right up there. The copy number is interpreted by the shift there. A blue bar tells you it's a gain, and I'll talk about the allele difference a little bit later. But in one shot, you know exactly where the gain is. Not only that, you can take the probe positions, go to the genome browser, and you know every single gene that is in that region. All we got by fish was the ones shown in red. So you know the breakpoints, you know how big it is, you know the size of it, and you know the genes that are there. So much more information. Ability to go right down to a gene is pretty impressive. GLUT1 deficiency syndrome, those of you that know Daryl DeVivo in pediatric neuro, he's the world expert on this, and I worked with him. 93% of his patients have mutations, but he had a small subset that by sequencing they couldn't see any mutation, and they were worried maybe there was a little microdeletion. So we ran his small group, and we got very excited. There you can see 3 of these patients had microdeletions. Chromosomes would be normal in these cases. A little bit disappointed about the twin girls in patients 6 and 7. The point of the slide is to show you resolution. This was run on our 500,000 probe array, When we moved to the next generation array that had, I think, 1.8 million, we decided to rerun these patients, and lo and behold, the twin girls show a microdeletion, as do patients 6 and 7, and when you zoom in, hovers right over the SLC2A1 gene, which is the gene implicated in GLUT1 deficiency syndrome. So just a nice example. We can go right down to a gene in certain cases when it's causative of a disease, and you really do need the probe coverage in order to detect those types of things. The last category is what I mentioned about the genotypes. And I know it may be a lot to think of, but by the time I'm finished, hopefully you'll get it. It's very visual and very easy. So we look at the allele difference, and I know that's already thrown people off of calculations of A minus B equals 0. But in the allele graph that I'm going to show you, think of the homozygous AAs are assigned to position 1, the heterozygous to position B, because A minus B equals 0, and BB is going to be at negative 1. Each allele by itself is 0.5, so the B's are negative, so minus 0.5 minus 0.5 is going to give you minus 1, and A is 0.5, 0.5, it's going to give you plus 1. And so if we look at this little diagram over here, the polymorphic SNPs are going to be the ones in color, and there you see the AAs at 1, the ABs at 0, and the BBs at negative 1. And if I fill that in with actual microarray data, that's what it looks like. So we get used to seeing these 3 characteristic tracks that represent 2 chromosomes when we see AA, AB, and BB. So we see those 3 tracks, we know there are 2 chromosomes. How's it helpful? Well, in uniparental disomy, that's when you have 2 identical copies or 2 copies from one parent. How does that occur? One of the primary ways we think it occurs is either by trisomy or monosomy rescue. If you have a conception that has 3 copies, in this case of chromosome 2, that would be lethal. If very early on it lost one of those copies, such that you left, in this case, 2 maternal copies, that would be uniparental disomy of maternal origin. If you have a single monosomy, again, that would be lethal. If very early on in embryonic development that duplicated, you get 2, in this case, copies, identical copies of the father. What would that look like on a SNP array? So that's showing you the normal 14 with the characteristic 3 tracks. But in isodisomy or uniparental disomy, you get complete absence of that middle track. Again, you don't have to be a cytogeneticist to know that. You just look at your SNPs and say, oh, where's that whole middle AB track? It's gone. That's a strong indication of uniparental disomy. And this is kind of nice because you see isodisomy with crossing over. You can see the exact points of crossing over. Why is that important? This is a normal individual. This is an individual that has Angelman syndrome. We know that UPD15 of maternal, let's see, paternal deletion gives you, of paternal origin gives you Angelman syndrome. So it's, you need to know, this is very important for certain regions that are imprinted, like Angelman syndrome and Prader-Willi syndrome. We also know that these LCSH, which is long contiguous stretches of homozygosity, when there is identity by descent, you do see this, these long contiguous stretches of homozygosity. If you come from the same parent or grandparent or great-grandparent, you're going to have these stretches that are identical, which would be LCSH regions. And that's just an example kind of shown there. But all of us have these little LCSH regions, anywhere from 0.25 to about 15 megabases. The average is much, much smaller, 0.25 to about 6. So when you get copy-neutral LCSH that's larger than 15 megabases on a single chromosome, we get concerned about uniparental disomy. When you see this on multiple chromosomes, then the indication is that there is consanguinity, identity by descent, which is important when we see in pediatric cases because often that unmasks a recessively inherited disease and manifests. This is what it looks like, consanguinity. The purple bars kind of represent these LCSH regions. Just 2 examples are shown for chromosome 4 and 17 on the right, but you'll see multiple LCSH regions across the genome. Very quick diagnosis of consanguinity. I don't know if it's a diagnosis, but an indication of consanguinity. Using the genotypes to ascertain parent of origin is important. Why? For certain imprinted diseases again, and we can do that. Here we would run the array on all parents, and over here we can actually see very easily that the proband only has the paternal genotype. It didn't inherit anything from the mother. So that means the loss comes from the maternal chromosome. Using the SNP to provide confirmation, again, we can look at certain deletions, and when we look at those tracks, again, we know the copy number. Remember I said when we have two chromosomes, we have the three characteristic tracks, AA, AB, and BB. When there's a deletion, you have one chromosome, so you can only be A or B. You can't be AB when you have one chromosome. And so you see these two tracks and this characteristic pinching in. Here you can see, again, in a patient with Prader-Willi, this pinching in. You see only two tracks over there. That's the log 2 dipping, and you can see a deletion. Allele graphs for trisomy, what does 3 copies look like? Well, there's disomy. We see the characteristic 3 copies. Trisomy, we're going to see 4 copies. Shift in the log 2 ratio, it's going to jump up. But because there are now 3 chromosomes, you get different genotypes, and those are all the possibilities, AAA, AAB, ABB, and BBB. So when we see these 4 genotypes, we know there are 3 copies of whatever that is. Again, a way of looking at the genotypes and knowing what the copy number is. This is just what it looks like for tetrasomy, again representing all the different genotypes. And I like this because it shows everything. This is where there is a copy number of 2. This is copy number of 3. And that's copy number of 4. Excuse me, 1, because there's only A and B. Okay? Detecting triploidy. The triploidy is a complete extra set. When you do array CGH or SNP arrays and you're looking at the copy number, it looks normal. So you cannot distinguish triploidy by doing array CGH or even looking at the copy number aspect of a SNP array, because the normalization basically cancels everything out. So the way you can pick that up is by looking at the SNPs. So this is a 69 XXX, copy number of 2 by the copy number algorithm. The log 2 doesn't shift, but when you look at the allele tracks on every single chromosome, you're going to see 4. You can tell whether it's a female or male by looking at the sex chromosomes. There, 3 copies tells you 2 X's and 1 Y. So in the last couple of minutes, I'm going to move quickly just to show you the applications in a prenatal setting. Actually, all this work led to a consensus statement by the ACMG about its use as a first-tier test over chromosomes compared to, in 2010. And then we asked this question here at Columbia, and I think Ron Wapner is going to talk to you about this in a lot more detail. Really, it's not the same. There's limits of ultrasound. That's how people use the information. There's very big differences between using this test for prenatal versus pediatric. And those are some of the reasons. And this led to the publication, and again, Ron will show you, which we did. It was a multi-center trial coming out of Columbia, but involving Baylor College of Medicine, Emory, Signature Genomics, and multiple clinical sites across the country. The most intriguing thing that we found is that about 1 in 60 individuals referred for what we would consider low risk for microdeletions and microduplications. Remember, microdeletions and microduplications are not age associated. Only aneuploidy is, full gains and losses of chromosomes. So we do not expect to see a difference for microdeletion incidence whether you are 21 or 37. And so what we found in what we would consider our low risk group for microdeletions, about 1 in 60 of them had a clinically significant copy number. And when you change the bias and look at ultrasound anomalies, about 1 in 17. So that was the most surprising finding, I think, from the prenatal microarray study. It's not all Down syndrome, and so we see the typical microdeletion or microduplication syndrome show up. We also see all the ones that we have been looking at and seen in many of the postnatal studies. Structural anomalies, so obviously improved counseling for structural anomalies, because about 6% of them will have a normal karyotype, but a clinically significant microarray finding, very similar to previous smaller studies. So when you see an ultrasound cardiac defect on ultrasound, in the past you could do FISH, but FISH would only account for 75% of DeGeorge cases, because only about 75 of them have cardiac defects. And it's not those that have cardiac defects, there's many other reasons. So if you saw a cardiac defect and did FISH, the percent that you would not detect would be about 67% of other reasons. So we see that now. These are some of the cases that lead to cardiac defects that are associated with congenital heart defects. This is the actual frequency of specific organ systems. So something going to be quite low, like abdominal wall defects, but some of the others are going to be pretty high. The overall average was 6%. But I think Ron published a paper last year, a really great paper, that actually shows you the frequencies of the CNVs according to specific organ abnormalities. Recurrent CNVs that have the potential to cause neurocognitive impairment. This occurred in about 0.8% or 1 in 125 cases that was sampled for advanced maternal age or positive screen. These CNVs are often associated with reduced penetrance and or variable expressivity. Do you guys know what that is? Have you had any lectures on that? Okay, this is an emerging, I shouldn't say emerging, but it's become prevalent with the fact of copy number changes. Penetrance refers to the likelihood that when you inherit something, you'll manifest it. So if the penetrance is 100% on an inherited mutation, it's not only for copy number variants, it could be for mutations or deletions or duplications. The penetrance is 100%. If I inherit that, that means I will manifest that disease. Variable expressivity refers to the fact of how severe I will manifest it. So certain diseases have variable expressivity which show a spectrum of the phenotype. Very often penetrance and variable expressivity are linked. So you can have 100% penetrance, but there you could still see variable expressivity. And what that means is that you can show a range of phenotypic findings. Some of the examples are NPL1 deletions and the 16p microdeletions and microduplications that have been implicated in autism and some other congenital abnormalities. So CNVs that confer a susceptibility for a spectrum of neurodevelopmental or congenital disorders. There's a lot of work being done on this, and what we see is in some cases, CNVs associated with the phenotypic heterogeneity may need, or the theory is, that it may need a second hit, which could be a mutation. It could be another copy number change. It could be an epigenetic change. We're still trying to figure out what these things are. An example that's at least postulated is the distal chromosome 22 microduplication may act as a susceptibility factor that requires a modifying factor or further challenge to the system for a clinical phenotype to emerge. And so that may have to do with penetrance or expressivity. Just because you inherit some of these things, we've seen, and we are referring to many of these as susceptibility factors rather than definitive associations. As we said, not all copy numbers are disease associated, and we spoke about these copy number changes. As I said to you, they can vary from 6% to 90% of any chromosome, the average of about 12 CNVs per person. That's using that 35K resolution, but that's significantly higher now with higher resolution studies, especially using next generation sequencing. Most of these are not associated with pathology. They're almost always inherited, and many contain known genes. And I think this is really part of our variation. What makes us all different are a lot of these copy number changes. How do we approach this? Obviously, we do clinical testing. We look to see, is it a new finding? Is it de novo? Is it something that's happened new in the child that you're looking at or the fetus that you're looking at? Obviously, you get more concerned for de novo findings, particularly if they're in a gene that is known to be clinically significant. So we look at gene content. We look at inheritance. We do a lot of comparison to other known cases, other big databases, the Toronto database of normal variants. In-house databases are very important. There's the International Standards for Cytogenetic Arrays. I think they've changed their name now. I don't remember what it is. And as I said, we look at content, etc. Just to show you how this is emerging, at the beginning of the prenatal study, we classified about 2.5% of variants of unknown significance and about 0.9% of pathogenic. Just before we published, we got all the directors together and said, okay, take these findings and using your knowledge in 2012, how would you classify these? And you can see that that dropped, the variants of unknown significance, we got a handle on a lot of them, that dropped down to 1.5%, Pathogenic went from 0.9 to 1.7, and actually we've been doing this every year, and that is slowly modifying every year. So we're getting better and better. There's so much more work that's been done for us to really do better when we see these variants of unknown significance. Looking at the frequency, these are some of the common cast of characters, but what I'll draw your attention to is the fact that over 50% of them were a single non-recurring occurrence. So you couldn't predict a priori, and this has relevance for the non-invasive prenatal testing, which I think hopefully at some stage you'll be learning about. It just means that you can put a kind of a small number of them over there, but more than 50% of the time you will not be picking up a microdeletion or microduplication region of interest. There's probably about 6 or 7 slides left. I'm just going to show you the utility in stillbirths as well as in products of conception. The biggest issue with these is the ability to get a result because of the delay from the fetal loss to the time they actually do the studies, as well as contamination from maternal tissue. And so the most obvious benefit of using a SNP array or microarrays is the ability to get a meaningful result, at least in the POC study that I showed you. We got normal failure rates of 10 to 40%, and we got a success rate of over 99%. So in the population that we would have looked at, there would have been 240 to 960 patients that would not have received a result. Then, more importantly, the ability to know whether or not what you're looking at is maternal. There's no point saying it's a normal female if you've just studied the mother and you don't get an understanding of why there was a fetal loss. And so the ability to actually discern is it maternal versus fetal is very, very important. And again, in the POC study, we prevented 539 women from obtaining a misleading normal female result. Again, we can go way beyond the threshold, and this is what we see for cytogenetics. And in some cases, we see a second cytogenetic aberration that would not have been seen by cytogenetics. And this is particularly important for unbalanced rearrangements, because if you see an unbalanced rearrangement that is not discerned, you need to look at the parents, because if one of the parents is a carrier, there are implications for future pregnancies. Again, significant increased PGD, as I mentioned. There's lots of ways that we do PGD, but really using the newer technologies, we are seeing improvements in outcome. And so looking at the PGD group versus control with respect to implantation rates, clinical pregnancy rates, when you split it out by age, it's pretty phenomenal. Women over the age of 35 are doing better than the controls after screening. And so what we're finding is that after more than 20 years, we're finally seeing the improved outcomes that were originally predicted that should occur by removal of the aneuploid embryos from the transfer pool. And this has really come from the development of the new technology.